In a forest along the Niagara Escarpment, a researcher crouches beside a young chestnut sapling. She reaches for a printout of its DNA. On that single page, spelled out in 4 molecular letters repeated millions of times, lies the definitive answer to whether this tree will survive or fall to the microscopic plague that wiped out over 4 billion of its kind across eastern Canada and the American borderlands. The sapling everyone swore would rescue this species was engineered in a lab, celebrated in newspapers, and funded with millions. It collapsed. The breakthrough actually bringing the chestnut back to our northern woodlands is older, slower, and stranger than anyone anticipated. To grasp why that DNA sequence matters, you have to understand what vanished. Go back 150 years into the hardwood stands of southern Ontario, stretching from Lake Erie to the Gulf of St. Lawrence. Roughly 1 out of every 4 canopy trees was an American chestnut. They dominated millions of hectares of eastern forest, some standing 10 stories high. In late autumn, they dropped massive cascades of nuts. Black bears, white-tailed deer, wild turkeys, and passenger pigeons lived off the harvest. Homesteaders turned livestock loose to fatten on the forest floor. The timber was virtually rot-resistant, used for barns, fences, and homes. Entire rural communities were sustained by this single species. Now walk through those exact same woods. The chestnut has essentially disappeared. Yet if you hike through the hills today, you can still find them. Fragile shoots barely 4 meters tall sprouting out of ancient stumps, bark scarred with orange lesions, dying before they flower. What survives is a ghost, a sick echo of a forest giant. The natural baseline was shattered, and we mistook the damaged remnant for normal. In 1904, orange pustules were noticed on chestnut bark in a public zoo. The killer was an invasive Asian fungus, Cryphonectria parasitica. It invades wounds in the bark and pumps out oxalic acid, burning fatal lesions called cankers. The lesion encircles the trunk, strangling the tree and killing the canopy above. Cruelly, the roots remain alive, firing up new sprouts that grow for a few years before getting infected and dying back again, trapped in an endless loop. It spread across the continent like wildfire, crossing the Niagara boundary within 2 decades. By 1950, 4 billion trees were dead, rendering the dominant canopy species functionally extinct. Forestry crews felled infected stands and cleared massive buffer zones, but the pathogen's microscopic spores rode the wind, rain, and wildlife across every barrier. The fungus could also harbor quietly in mature red oaks, waiting. You cannot quarantine the wind. Every direct attack failed. Decades later, biotechnology promised a silver bullet. Geneticists proposed a question. What if you don't fight the fungus, but disarm its weapon? Wheat possesses a gene producing oxalate oxidase, an enzyme that breaks down oxalic acid into harmless molecules. Researchers inserted that wheat gene directly into the chestnut genome, creating a line named Darling 58. It was pitched as a groundbreaking conservation triumph and backed by millions in funding. Then, in late 2023, support for Darling 58 was abruptly withdrawn. In field trials, the trees grew slower, blight resistance proved inconsistent, and overall survival was poor. Worse, researchers discovered a disastrous mix-up. Due to greenhouse mislabeling years earlier, the test trees were actually a sister line, Darling 54, which carried a hidden genetic defect that compromised its health. The flagship of forest genetic engineering had been the wrong tree for nearly a decade. Beyond the error lay a deeper lesson. A single inserted gene was never going to be enough. Asian chestnut species co-evolved with the blight over millions of years, simply scarring over when attacked. Traditional breeders had long tried backcrossing, crossing native chestnuts with Chinese chestnuts to borrow resistance, then repeatedly breeding offspring back to native trees to dilute out Asian growth traits. The goal was a tree that grew tall and straight while retaining an Asian defense shield. But it took a century without reaching the finish line. A landmark study in Science revealed why. Resistance is not governed by a single gene, but distributed across dozens of genes working in tandem. It is a multi-layered immune system built over eons. You cannot replicate that evolutionary history by stitching a single crop gene onto a wild tree. Backcrossing ran into a massive bottleneck. With resistance scattered across dozens of genes, every hybrid seedling was a random roll of the genetic dice. Most inherited weak combinations. To find the rare winners, breeders had to plant thousands of saplings, wait 5 to 7 years, manually slice open their bark to insert live blight fungus, and wait years more to see which survived. Because chestnuts take nearly a decade to mature, every cycle was a 10-year gamble conducted in the dark. Genomic sequencing solved this invisible problem. Scientists mapped the genomes of native and Asian chestnuts, matching genetic patterns against decades of real-world survival data. Through genomic selection, researchers can now analyze DNA from a 2-week-old seedling and predict its future resistance immediately, compressing decades of field testing into days. Using DNA-guided selection, the upcoming generation of chestnuts is projected to possess double the blight resistance of previous lines, while remaining about 75% Native American chestnut. That proportion retains the towering height, rapid growth, and canopy competitiveness of the native species, carrying just enough borrowed code to survive. No one synthetic engineered this defense. Scientists simply learned to read the genetic script and select seedlings that already inherited it. In research plots across the eastern hardwoods, 13-year-old backcross hybrids stand tall and green despite fungal spores in the air. Living proof that a mostly native chestnut can wall off infection and keep climbing. While scattered ancient survivors exist in the wild, genetic testing shows they pass on only modest resistance. They cannot rescue the species alone. The story takes another turn in Europe, where the same fungus invaded sweet chestnut forests. There, the woods began healing naturally, as cankers transformed into shallow scars. The cause was Cryphonectria hypovirus 1, a hyperparasitic virus that infects the fungus itself, drastically reducing its virulence so trees can easily wall off lesions. In Europe, fungal populations were genetically uniform, allowing colonies to fuse via hyphal anastomosis and spread the healing virus naturally across entire regions. When North American scientists tried importing the hypovirus, it failed. Multiple Asian importations had blanketed North American woods with dozens of genetically incompatible fungal strains. When incompatible strains touch, cell boundaries undergo rapid cell death rather than fusing, creating a biological wall that blocked the virus. This contrast proved that nature rarely offers uniform remedies. Neither an imported virus nor a single inserted gene could save our woodlands. The solution required tapping into the layered genetic defenses forged over evolutionary history. Today, across test plots in southern Ontario and eastern Canada, genomic-guided chestnuts carrying roughly 75% native DNA are establishing strong roots. Seed orchards using genomic selection are projected to produce wild-ready seed for active reforestation within the next decade, offering self-sustaining populations for provincial parks and private woodlots. Restoring the chestnut rebuilds a severed ecological web. The loss of a dependable autumn nut crop forced black bears, deer, wild turkeys, and rodents to rely on inconsistent acorn mast. Reintroducing the canopy restores a stable food foundation. Historically, rot-resistant chestnut timber also provided rural communities with durable lumber without chemical preservatives. The chestnut's return offers a vital lesson for managing modern threats, like emerald ash borer or Dutch elm disease. Lasting solutions require understanding evolutionary biology and genetic diversity, rather than chasing short-term technical fixes. Standing along the Niagara Escarpment, watching a young chestnut reach toward the sky, we are witnessing the return of a lost landscape. After a century trapped as a scrubby sprout in the understory, the giant of the eastern forest is stepping back into the canopy, brought home by reading the complex answers nature wrote inside its code.